So we are here today at the Biomedical Imaging Center with Professor Brad Sutton. This is a phenomenal facility at the University of Illinois that's doing cutting-edge research using MRIs. Can you tell us a bit about what you do here?
Yep, so we have a couple of different MRI scanners. We have 2 human 3 Tesla scanners, and then we have an animal 14.1 Tesla scanner. And the Tesla is the magnetic field strength, and so the higher the strength, the better the signal you can get, and the more spatial resolution then you can get from those images. We have a variety of other supporting equipment too. So we have optical imaging, we have a PET spec CT animal scanner, we have ultrasound system, and we're soon to have a bioluminescence scanner too.
Wow.
So a one-stop shop for seeing how brain changes, how body function changes, for putting different types of contrast agents into animals and humans.
Okay, well let me see if I got this right. So the way MRI, which is magnetic resonant imaging, works is that you have an axis of the hydrogen nuclei, right? Mm-hmm. And if you basically vibrate it the right way, it will process. And therefore it gives you a signal you can pick up.
Right.
Now, the only thing that's key about this is whenever you have biologic activity, like you're thinking a certain thing, your brain, that area of your brain uses more sugar, has more metabolism going on, and therefore it has more of these free hydrogen radicals and hydrogen ions, and the signal's bigger.
Yep, so the way functional MRI works, which is a large part of what we do here, so we want to see how the brain functions in different situations, and we want to actually see if we can change that in some cases. So as you get older, your brain may change the way it's functioning, it changes its structure, and we do a couple of different interventions here to try to change that. So we look at structure and function for that. The way functional MRI works is that when your brain is processing a certain task, you have hemoglobin carrying oxygen to the tissues. Hemoglobin has iron in it, and iron in a magnetic field has a very strong effect.
So you don't just look at the hydrogen, you can look at the iron?
So we're looking at the hydrogens around the iron. They get impacted by the disruption to the magnetic field. And so what happens is if hemoglobin comes along, it has a lot of oxygen on it, the oxygen is shielding the iron from the magnetic field, and so the hydrogens around there have a nice uniform magnetic field. But when the brain is active, it gives up the oxygen, and that causes then the iron to be free to disrupt the magnetic field. It disrupts the signal then from the hydrogens that are nearby.
Wow.
So.
And of course, this is all non-invasive. I mean, as we'll see in the machine, you sit there, you don't notice anything, and you can see the brain actually working. So you can ask someone a question, and you can see what part of their brain lights up.
Right, right. And we have a couple of examples too on the slides here if you want to see of that functional type task. Let me just go to the slide here. So this is an example here of different function across the brain in older adults with a physical fitness intervention. So trying to look to see how activation in different parts of the brain, and particularly for older people, we're interested in things like executive function, the ability to focus in on a particular task and not be distracted by other tasks. That happens in the front of the brain, and so we're looking at various slices here in the brain. This is a side view. And so we have an investigator here on campus, Art Kramer, who's director of Beckman Institute, doing work on how physical fitness impacts these things. He's doing it in older adults and in kids too.
Okay. Yep. Well, that's great. And it's interesting, the slices. So the nice thing, just like one of those CT scans, right, you can actually section off by cross-correlating where the signal's coming from like that. Right? Just electronically, you can take those images and you can make all these different slices.
So it's a very flexible technology. We can choose any orientation you want. We can choose a slice and just look at that one slice and pull that slice out.
What kind of resolution do you get?
So for the human systems that we have here, typically we're looking at something about 1 millimeter, slightly less than 1 millimeter isotropic resolution. For the animal systems, we can get something more on the order of tens of microns for an animal. So so there's a lot of...
And that's because it's in the 14 Tesla.
It's in the 14 Tesla higher magnetic field.
Wow. Wow.
So
these are the same levels of magnetic fields we use in fusion energy, by the way.
All right, that's good.
Mostly because that's about as high a magnetic field you can get.
That's right, that's right. So, and you know, the human systems are going to higher fields too. So 7 Tesla, there's a 9.4 Tesla in University of Illinois, Chicago, and hopefully an 11.74 in Europe, and a 10.5, a couple 10.5s in the US.
Now, to get these high magnetic fields, these have to be superconducting magnets.
Yep.
Right. So the magnet is cooled down near liquid helium temperatures, and once that magnet has its field, it's a superconductor, so it always stays. There's no resistance to the current. until, of course, something goes wrong.
Yep, yep.
Yeah.
So when we come in, when we check on the magnets, we listen, and with the door open, you can hear the cryogen recycling pump, because the cryogens are constantly boiling off, and they get recondensed and put back
right.
to keep the temperature of the system at superconducting.
And so what happens if the magnet warms up?
So if it warms up too much, you'll get what's called a quench, and what will happen then is all that liquid will turn into a gas, and hopefully will be vented out of the building, probably over the street out here. Right. But then you had a superconductor because you had no resistance in the wires, and so now with the loss of superconducting, you get a lot of resistance and a lot of heat generated.
And so the wires might melt.
They could melt. Nowadays, the systems are quite a bit better that a quench doesn't necessarily mean that you need a new magnet. So
good.
They are much more robust these days.
Excellent.
So one of these maps here is looking at all the fiber bundles connecting different regions in the brain. So we can look and see for a proton in water that's sitting inside an axon. It can diffuse along that axon, but it can't get outside the walls of that axon very easy. And so by looking to see which direction water can diffuse, we can make a map of these fiber bundles. Wow. And then one other thing we've been doing lately too is we've been shaking people's heads in the scanner. So we put you on a pillow, and we shake you at about 50 hertz. Your head moves on the order of tens of microns, but your brain is going to move less than 10 microns. And we look to see how the shear waves propagate in the tissue, very small displacements, and then we can actually get a stiffness map out of your brain, and we can see regions of the brain that are tightly bundled fibers, such as here. This is the corpus callosum. You get tightly bundled fibers, and you get really bright stiffness map. Whereas regions on the outside, we may have fibers that are intermixed with each other, and they aren't as stiff, and they might actually be able to move against each other. So we can also measure viscosity too. And then we can do things with dynamics too. And so we can actually see some very fast movement of things, of protons. And so we've been doing a lot of work on making MRI very fast. So this is a Christmas song, but we can actually get speeds that rival any of the other imaging modalities. Typically, I'll stop the audio. If you're interested, Beckman Institute has a YouTube channel, and we have that up there. You can watch the whole thing.
So what parts of your brain light up when you sing Christmas carols?
So we can do that study. We can image function while we image dynamic too. But you would have language centers, you have listening centers, and things like that too. This we're actually interested in how the tongue is moving and how it's creating the sounds. So we're studying from healthy people, from older people, and how function changes with age, to cleft palate individuals that are born with disruptions to some of the muscles involved in motion too. So that's the primary driver for the study, is to see how, when some of those muscles are disrupted, how function occurs. So,
wow, pretty cool.
So great.
Should we see the machine?
Yep, that sounds great.
So magnetic fields fall off dramatically, especially for solenoid.
Yep.
What do you feel?
You got 12 type of
Yeah, so the 5 Gauss line is usually what people are worried about with regards to safety, and the 5 Gauss line runs through the wall in this room.
5 Gauss is nothing.
Yeah. So pacemakers and a lot of the other things, that's where we would limit your access. So yeah, the field does increase very quickly, like you said, it falls off pretty quickly. So I have in this block a roofing nail, okay, and it's taped in there very well, but you can see then how it gets sucked in. So yeah, so and then when you reach in to grab something like that too, all the wiring in your head, all your neurons and stuff, you have to be careful, because as you're reaching in, you're cutting all these magnetic field lines, and you're inducing currents in your head too. So people can get dizzy. If you were just to stick your head in really quickly and grab that back, you would get pretty dizzy. So would you like to throw that?
Yeah.
All right.
Yeah,
you can feel it too. Yeah, it starts to feel it.
You map out the field lines.
Yeah, you can feel it tries to align with the field lines. Yeah. Yeah, you can. Oh,
I was just going to see it.
So it's not enough out
Whoa,
there.
there we go.
Right here, it gets sucked in.
So I've always had fun with magnetic fields, being in fusion, right, and also having these big clubs. Now, I don't know if you know, we have a tokamak accelerator here on campus.
All right.
It'll get up to 1 Tesla.
All right.
That's big, right?
Yeah,
it's a whole bunch of these coils, non-superconducting. And I remember through grad school and through other things, right, especially talking with some of the old timers, who would say, oh yeah, we know when the field pulses to a certain level because their whole field of vision turned green.
All right.
I was
Yeah.
like,
Yeah.
I somehow don't think that
Yeah.
was really healthy.
That's right. So there are 3 safety issues associated with MRI scanners with people, and that would be one of them is we can only switch the magnetic fields. In order to image, we have to change the magnetic field in different portions of your brain in order to see things that are processing, as you said before,
Right.
at a different rate on one side of the brain versus the other, but we have to switch these fields pretty quickly. So the rate at which we switch those fields is limited so that we don't induce currents in people and they don't get sensations or an extra heartbeat or something like that.
Yeah.
And so that's one of them. The other one is the static field. If you have metal in you, you can't get in the magnet. And then the third one is heating. We are putting energy in to cause the
Cause the change, because of the thing you're seeing.
cause the
right?
things to actually spin off axis and process. And so we have to limit how much energy we put in so we don't heat you up. One other thing too, this is aluminum, okay? And so aluminum is not attracted to the magnetic field, okay? But inside the magnetic field, it does something very strange. So it does not operate how you would expect it to. And so it turns out that it's experiencing that magnetic field too, and that's inducing currents in the metal. And those currents are creating magnetic fields that oppose the change in magnetic field. Oops. So as it's trying to cut those lines, it actually slows down. And if you were to hold on to this, you can actually feel that as you twist your hand in there.
Oh yeah, Lenz's law. Even though it's not it's not magnetic, but it would definitely have the eddy currents.
Yeah.
Wow. That is really neat. It's like,
And if you have no metal in you or no concern, we can have you do that. You have zipper on your coat and stuff, but should be okay. But if you guys
Anyone else
wanted to
want to
try
try
that out, yeah.
yeah.
Until it
so
got
so
inside,
even my large cobalt chrome titanium pieces, I
you would
feel
still
that.
feel that.
Yeah, I don't want to.
And when they're imaging you too, those cause really big disruptions to the magnetic field.
Sure.
So there's a whole field of having to develop sequences, special ways to acquire data that will allow us to see close to those types of
Anyone
disruptions.
else?
And now that we're in here too, you can hear the compressor.
Yep.
Yeah.
Oh, I thought it was just nice, a good disco drum
It's a
beat,
good
right?
disco drum beat. If you don't hear the compressor, you would get worried because we would start boiling off cryogens. It would take a couple of days for us to boil off enough that we'd have a quench issue. But the room is completely encased in copper. We came through a door that's sealed when we came in. You can hear the air pump. And the window behind you has a mesh grid in it. And that's because we're listening for signals at 128 megahertz. The protons in your body are going to be creating this signal at 128 megahertz. There's a lot of 128 megahertz out there.
Radio.
And so we have the room has 100 dB attenuation in order to keep those signals out. And then everything being passed into the room has to go through a filter, or it has to go through a fiber optics through a port in the wall, so that we don't bring any of those signals in from outside. We have lots of coils that people put on. So this, you would get in here, you put your head in, and we'd snap the coil over you. Coils are a big issue. So MRI is traditionally a slow imaging modality where we can only image one sample in data space at a time, and our speed to travel through data space is limited by not creating currents in your brain, how fast we can switch the gradients around. So what people have done is they've developed channels with lots of individual elements. And so this coil has 12 elements around it. Each one of those 12 elements only sees a small part of your brain. And so now we only have to encode a much smaller amount of your brain in order to get a full image. But we have to stitch all those different coil images back together in order to get a good image. So this one has 12. We have another head coil that has 32 channels. And there are 64 and 128 channel versions now available too. So people are really trying to increase. The more elements you get, the smaller they get, and their penetration depth gets less. And so you could have an issue of getting good signal in the center of the brain if you had, say, a 128 channel coil with very small coil elements. So, and then we have coils for the knees, we have coils for the heart, we have special coils for everything so that you can get the best signal that you can get with something that's very localized to just the anatomy that you're interested in imaging. So,
Fascinating.
Yep, and then one other thing to say about that too, just to, this is a fact that people usually find interesting too. So the magnets cost about 2 to 3 million for a 3 Tesla scanner. There are cryogen costs each year too, which are quite a lot. There's maintenance contracts which are quite expensive too. Coils cost anywhere from, say, 40 or 50,000 up to 100,000 a piece also. We have special fiber optic presentation systems here in order to get signals in for the subject. We have a 47-inch LCD TV in the back of the room to give people a really high-resolution picture of what they're watching, but you can't just put a regular TV in the room. It's a very special TV too. So these things generally cost at least 10 times more than the non-MRI version, because they had to pull out all the ferrous components. They had to make sure it doesn't leak RF into the room. And usually they end up being not necessarily one of a kind, but a few of a kind systems. So,
yeah. Large research budget.
That's
and
right.
then who funds this effort?
So our money comes from a couple of different sources. We have a lot of investigators funded by NIH. We also have IARPA, which is the intelligence agency version of DARPA. We have Abbott Nutrition through the Center for Nutrition, Learning, and Memory. And then we have a variety of other smaller grants too, funding things here too.
Now, Illinois is real special in the MRI world, right?
Yeah.
Because it was invented here.
Yep.
Nobel Prize was
Nobel Prize 2003 to Paul Lauterbur for invention of MRI.
Okay.
And so he did that work just before he came here, but a lot of the early techniques that MRI used were developed by him. And in fact, to this day, we're still doing a couple of projects that are things that he started early on. The equipment couldn't actually do the experiment back then, and now we can. The equipment can actually do these things. So it's really great to have a lot of this history around to look at, because you think you thought of something new and you find out that it's not new. Yep, we're in the control room for this, and so you can see on the magnet it says 600/89, and the 600 is the megahertz. So the 3 Tesla scanner was at 128 megahertz, and so this one's at 600 megahertz. And so 14.1 Tesla, you can get much better resolution, and there's a couple posters on the wall you can look at as we go out to see. But the gradients are also much stronger here, which allow us to encode spatial position. So I was saying we can only switch the magnetic field so fast on a human system. Well, here the distance is a lot smaller for a mouse, so we have much stronger things that change the magnetic field.
And the tube is basically, what, about that diameter?
Yeah, so the 89 on there indicates the diameter of the tube with nothing else in it. And so we have gradients and we have an RF coil in there.
Mm-hmm.
And so in the end, we're left with
And that's millimeters, right?
Yeah, in the end, we're left with 2.5 centimeters to image with.
That's
and
1
so
inch.
if you have an overweight mouse, he will not fit.
That's right.
So sometimes we end up scanning samples of things instead of maybe whole animals. But we do a lot of mice here too. And so and the sequences you run with an animal, usually they're anesthetized, and so they're able to maintain a little bit longer scan time than the humans. Humans, we try to get in and out of the system in an hour and a half, but we're doing lots of different scans on them, and we're interacting with them and making sure everybody's fine and they can get out at any time they want. With the animal, we're able to scan for maybe an hour or two under anesthesia just fine, and we usually are running maybe just one scan that we're really trying to get a nice structural scan from them or something like that.
The mice don't complain too much, right?
Yeah, they don't complain.
Okay.
They don't ask to get out.
Okay.
Say they have to use the restroom.
Yeah.
So.
Yeah, I mean, it's very, very important role that this type of animal research does. And it's good for the world to know, no, we're not trying to torture little tiny animals, right? What we're trying to do is help humankind. And you can do that so much more effectively when you have control over the breeding, the diet, the exercise, the entire life cycle of a particular animal. You don't hurt them, right? But it is a valuable tool. After all, if you think about it, 98% of the world are not vegetarians, right? So we also raise animals to eat. So why not raise animals to be able to try to cure disease and to learn things about and so forth?
Do you have any questions on this system? There's a couple of images
Yeah, I
out
see
in the
some of
hallway.
these images. They're really cool.
So we have imaged everything from, mice, of course, and there's some mouse images on this poster over here. This is a good example of how to use an animal model. There's an aging and exercise study on humans where we know people who engage in exercise over a shorter period of time, 6 months, we can actually measure increases in brain volume in those people relative to sedentary people over the same 6 months. So think about that as you go, that it doesn't take much time in order to have an effect. But we wonder what's actually causing that effect. Is it new neurons? Is it new blood flow? And so in order to get that, we need to actually do an animal model where we have the animals get the same effect. And so for this, we've been trying to match the acquisition sequence, trying to get similar contrast in the animal model so that we can actually go in and see what exactly is changing in the brain, whereas you couldn't do that with the humans.
So what's the answer? What's
So ongoing project still. So we'll let you know in another year or two. Yeah.
Okay.
So, but you have the ability with animals to stain where new neurons are coming from. You can stain blood vessels, and you can look and see, are new neurons being formed? At what rate are they being formed in the exercise animals as opposed to the control animals? So.
Wow. And
We've
here's your mouse in the tube diagram.
In the tube.
Okay.
Yep. And
Okay.
you have to keep them warm, and so you
Yeah.
have to route heat. There's just not a lot of space, and so
Right.
you have to figure out how to get everything in there: gas anesthesia, heat, and also get the RF coil in there and position the brain where you want it. So.
Yeah, and the mouse probably doesn't like cooperate. Like, hey, move to your left.
That's right. That's right. Can't tell them to move around.
That's right.
So, so I think, other things on the wall here. We've imaged an ant head. We have tadpoles. We have cornworms. There were cornworm parasites. There were parasites that we were able to image coming out of a worm and to see the process of how they damage the worms. We've done fish, so there's a stickleback fish. So lots of investigators on campus looking at various aspects of things, and these are either the actual thing they're hoping to image, or these are animal models for some other human disorder.
That's great.
You get structure, you get protons, you can see where they are, so you can see an image. We can get function because you can look at things like hemoglobin and see if it's shielded by oxygen or not. We can get diffusion, we showed you that. You can see what's restricted in the brain. We can get motion, we can shake people's brains and get motion, or we can watch motion happen. We can look at blood flow, that's another type of motion, just look to see where blood is going. We can also get metabolic information. You can see a couple of different chemical species in the brain and what their concentrations are. So one way we monitor health of tissue is we might look for a marker of healthy neurons in the brain that we can actually see the concentration of that marker. But there's a lot of different contrasts available and image information available from MRI. It's not just, in some modalities you have attenuation of a radiation source going through an object, and you're a little bit limited on what you can interpret. MRI, we get lots of different signals coming back from those protons.
Yeah, it's really an amazing thing, and thank you so much for letting us into your center today,
Great.
learning all this cool stuff, and
Great.
that's what you need to know about MRIs.